be honest.
Somebody's mic is on.
Good afternoon, friends. Thank you for joining us from different places across the country. It's a request to keep your microphones muted and videos in off mode during the talk. Keep your questions,
Be
if
honest.
any, ready in the chat box for the
Somebody's
question
mic is
answer
on.
session planned at the end. Friends. We would
Thank you
like
for
to
joining
have your
us from
suggestions
different
and
places
comments.
across the country.
For the same, a link for feedback form would be available in your respective WhatsApp groups. The link would be open till 6 in the evening. You will have to wait for a few days for your participation certificates, which are linked to the feedback form. Please take out some time to fill it up. Thank you.
हाँ बोल ना तिथं नाही बंद आहे
I'm admitting them.
So I just put that.
Just announce. Good afternoon. Participants who are unable to join us on Teams may please be with us on YouTube where we are streaming live. The link for the same is shared in your WhatsApp groups. I repeat, please join us on YouTube. We are streaming live, and for the same you can have the link in your WhatsApp group. Good afternoon, friends. Thank you for joining us from different places across the country. It's a request to keep your microphones muted and videos in off mode during the talk. Keep your questions, if any, ready in the chat box for the question answer session planned at the end. We would like to have your suggestion and comments. For the same, link for feedback form would be available in the respective WhatsApp groups. The link would be open till 6 in the evening. You will have to wait for a few days' time for your participation certificates, which are linked to feedback form. Thank you for joining us. And those participants who cannot join us on Teams may please be with us on YouTube. We are streaming live, and the link for the same is provided in the respective WhatsApp group. Thank you.
Good afternoon to one and all. I'm Srishti Chitte and I'm a BSc Century student. Good afternoon to one and all. I'm Srishti Chitte and I'm a BSc Century student. On the behalf of Hislop School of Biotechnology and the Department of Chemistry, Hislop College, I welcome you all to this webinar. Today, we have a diverse audience from Kerala to Punjab, which includes undergraduate students, postgraduate students, research scholars, and faculty members. Today is the day when science is appreciated and honored all over the world. Yes, I'm talking about the International Science Day, also known as the World Science Day for Peace and Development. The aim of celebrating this day is to promote scientific exchange among countries, to empower the scientific community, and to acknowledge the importance of science in our lives. Before we begin with the webinar, I invite Karen for the opening prayer.
Thank you, Srishti. Let us bow our heads in prayer. Heavenly Father, we come to you today asking for your guidance, wisdom, and support as we embark on this quest for knowledge. Lord, I place unto thy hands the college officials and our guest speaker. Grant them your power and wisdom as they impart knowledge. Father, Bless the students gathered here today that they may imbibe the erudition gained today. Lord, I pray for the entire world in these trying times. Bless and protect each and every one of us. Grant us the power to fight through this pandemic and emerge victorious. In Jesus' name we pray. Amen.
Thank you, Karen. I would now request Dr. Prateek Michael, Vice Principal and Head of Chemistry Department, for the introductory remarks. Sir, please.
Thank you, Srishti. For the webinar, the speaker for today, Professor Deepshika Chakraborty, faculty members from invited institutions, dear participants, and my fellow HISLOP members. Namaste, good afternoon, and greetings to you all. On behalf of the HISLOP Education Society, the College Management, Dr. Prashant Sherke, our principal, the two organizing departments, HISLOP School of Biotechnology and Department of Chemistry, the entire HISLOP family, and on my personal behalf, I extend a very cordial welcome first to Professor Deepshika Chakraborty, faculty participants, and to all dear young participants of today's webinar, Expanding Horizons, and for the talk on why bacteria wants to live in a vacuole. We at HISLOP are so pleased and excited to have our very own Nagpurian, Professor Deepshika, with us today. I want to thank you for sparing your time and being with us to celebrate the International Science Day and to edify the student. Come January 2020, and we would not have even thought of being on a platform together for an interaction like today. But today we have faculty members and students joining in from different parts and some Hislopians from different parts of the globe as well. Another webinar, as one would think, but we are all here with HISLOP, a 137-year young college, having distinction to be institution in a heritage status college. Today, it's an important day for all of us and the scientific community worldwide to arise together in celebrating International Science Day or the World Science Day for Peace and Development, as called by UNESCO. The day offers opportunity to highlight the role of science to society, the relevance of science in our daily life, and engaging and creating awareness about emerging scientific issues, both at local and global level, and keeping the citizens well informed. The day offers an opportunity to mobilize all actors around the topic of science for peace and development, from government officials to media to social people. And the world is struggling with COVID pandemic. The world focuses of World Science Day is on science for and with society in dealing with the global pandemic. Just before I close, let me give you a scenario. We have homes with very restricted movements, and many of us have not even visited our places of worship. Does it mean we have not been able to connect or worship the Almighty? Absolutely not. It is not imperative to worship or celebrate in life. Instead, we need to be responsible, maintain physical distancing, yet connect socially. Each life is truly a gift of God. We can honor the gift by cherishing our own lives as well as respecting and the light. We can share the light and truth with the world around us. Hence, commitment of science is for peace and development. I eagerly look forward to the talk and wish today's webinar is going to be a beneficial and an informative one for one and all. Thank you and have a great time.
Thank you, sir. I now invite Dr. Sunita Bundale, Director, Hislop School of Biology, to introduce the speaker to the audience.
Thank you, Srishti, and thank you all for joining us today. It gives me immense pleasure to introduce the speaker of the day, Professor Deepshikha Chakravarty, Head of the Department Microbiology and Cell Biology at the Indian Institute of Science, Bangalore. She completed bachelor's and master's from Nagpur University and then went on to complete her PhD in biotechnology at NCCS Pune. She performed her postdoctoral studies in Japan and Germany. Her research mainly revolves around infectious diseases and host pathogen interactions with a particular interest in the pathogen Salmonella, responsible for causing typhoid. Her research has resulted in more than 135 publications in international peer-reviewed journals and chapters in biomedical books, as well as patents related to Salmonella diagnosis and vaccines. Besides her research, she teaches graduate students at university, actively participates in national and international committees and societies, and serves as editor and board member for several journals. During the course of her career, Professor Chakravarty has received several honors and awards, including the Astra Chair Professorship in 2018, Fellowship for National Academy of Sciences India in 2012, Reliance Platinum Jubilee Award in 2015, DAE SRC Outstanding Investigator Award in 2015, National Bioscience Award by Department of Biotech in 2010, the ASM Indo-USA Professorship in 2005, Humboldt Fellowship Award Germany, and Daiichi Foundation Fellowship for Research in Japan, just to name a few. So without much delay, I would like to invite Professor Deepshikha to deliver the much awaited talk to diverse participants from various universities and research institutes from all over India. Over to you, Deepshikha.
Yeah, thank you, Dr. Sunita. It's very interesting to call my one of my very best friend. We were classmates in MSc, and it is really my great pleasure. So let me first begin by thanking my wonderful colleagues from Hislop College. I had been to the Hislop College before when Sunita invited me. I have interacted with very vibrant young colleagues, graduate colleagues. And I would like to thank also Dr. Michael, HOD Chemistry, for beginning the seminar, and all the participants, all my very young colleagues. Otherwise, I would have been there and we would have had a wonderful discussion and wonderful, interactive sessions. But as the time is, and as the time is different, I am going to take you through this online mode. But having said that, let us understand that it is all about understanding that wavelength among each of us. And though you are all in different part of the country, I can still feel that, I'm just right in front of you delivering the talk. So I would request, in case Sunita, you are not hearing my voice or my slide, then Sunita, you can just unmute and talk to me and let me know. Now,
Yes, sure, Deepshika.
I'm fine, right?
Yeah.
Yeah, great.
We can hear you.
Yeah, great. So, and I will take the question in the end. So then I will open the chat box and let us have a very wonderful discussion. So thank you, Sunita, for this wonderful opportunity. I cannot thank her enough, because it is just not the friendship that we carry. And now I know that, how important the friends are all over, even to have these such a amazing scientific network and the connection. Me being from Nagpur also gives me a very important feeling to talk to all of you, because I always realize that Nagpur is one of the very happening city. It is one of the very prominent place where everything happens, right from the science to the, the literature, the art, as well as the politics. So this is, I am very proud to be a Nagpurian. And so let us begin our talk now. So I would request you, all my young colleagues, that you can put from your end the slide entire full screen mode, because looks like that there is a bit of an issue from my end if I try to project it in the full screen mode. So you can see my first slide.
Not yet, Deepshika.
The same thing which I have. 1 minute. Okay. 1 minute, I'm sharing now. Okay, can you see now?
Yes, we can see.
Great. Okay. So all of you, my young colleagues, kindly put your own screen into the full screen mode. You will see the 3 dots, and then press onto that and go to the full screen mode, whereas I am from my end will get into this mode only. Otherwise, I would have loved to have your questions in between, but looks like that in an online mode, it is good to have the questions where I will take all your questions one by one. We have time, and let us take me through this wonderful journey of life in a vacuole. Now we are kind of really mimicking the situation of a life in a vacuole. We have become, we ourselves are now kind of a homebound. So we are in our own vacuole, and this vacuole is really very interesting. I will also tell you that how bacteria loves to live in a vacuole, and why certain bacteria loves to live in a vacuole, what the vacuole gives the protection to them, and what are the strategies that they understand to live in a vacuole. So if you look into the research that my lab carries out, so we are a lab of roughly around now 14 to 15 years. I joined my IISc in 2004 as an assistant professor, and had a very wonderful group, my young colleagues, and from also very, very good trainees, which Sunita had sent me over years from Nagpur. Had one of the very successful student of Sunita, Dr. Akshay Date, who in fact graduated with me and is doing a wonderful work. So I would really thank Nagpur and Hislop College for providing us with such an amazing minds that can add up to the science that our lab is doing. So now our lab looks into how a bacterial pathogen bridges the gap. That is, we look into the metabolism, the ecology, and the therapeutics, how to intervene them, finally understanding the pathogenesis. So pathogenesis was the key for us to understand that how bacterial pathogen behave and why do they become a pathogen. It is like a success story of a mitochondria. Why not every bacteria can have a success story like we have in a mitochondria or as a chloroplast or plastid? Why there should be a tug of war between the bacteria and the host so that they will now get into this mode of so-called making us sick. So this is something which is a very interesting mindset of the bacterial community behavior, and something to do from the host side that led to the entire area of establishment of the pathogenesis. So indeed, 1 to 2% of the bacteria causes pathogenesis and they make us sick. And why do they cause? This is a very important question. So let me tell you in brief about our model organism, that is a Salmonella. Why Salmonella is so interesting? Now, Salmonella, whenever I tell you about Salmonella, and many of you are a student of microbiology, the first, hello? Hello? Okay, I thought there is some question. So the moment I say Salmonella, the first very important story that comes to your mind is Mary Mallon, and she was known as a typhoid Mary. So she was supposed to be one of the very important carrier women who never suffered from typhoid herself, but spread the typhoid to a variety and to several of the people where she worked. So she worked as a cook, and she spread that typhoid through her cooking because it was she harbored it under the nails, and whenever she used to cook, she had spread it. So Anthony Bourdain had written a very beautiful book on the Mary Mallon, and there were so many stories about her story of isolation. Later, she was isolated in the Long Island. Now, Salmonella is an extremely interesting and intelligent pathogen. The cause of infection by Salmonella is through the orophycal route. That means the Salmonella gets inside the body through contaminated food and water. And it is a peritrichous in nature. It's a gram-negative peritrichous bacteria, and it enters the human body through contaminated food and water. Now imagine that the Salmonella travels all the way through to the small intestine via passing the stomach, and the stomach has an acidic pH of roughly around 2.3 to 2.5, and it survives. Now how Salmonella survives this acidic stress in the stomach is through their very interesting two-component system, which gives them a protection against this acidic stress. When it lands up in the small intestine, there is a region in the small intestine which is known as a Peyer's patches, P-A-Y-A-R, Peyer's patches. Now, Peyer is a name of a scientist who discovered these Peyer's patches. So these are a kind of a small, not known yet, but it is a very interesting cell because through this cell, the bacteria takes a niche and it translocates. Now, you will ask me a question that how is it possible that the pathogen can at all invade these deep tissues? How is it possible? Because on the top of these small intestine enterocytes, you call them as enterocyte, epithelial cell, lay a very thick mucus, which is around 100 micron. And on top of these 100 micron mucus are the gut flora. So 230,000 different species of the gut flora exist there. So how is it possible that you have such a bacteria like Salmonella, which can cross across this thick mucous membrane through fight amongst the gut flora and actually reach the underlying layer? Look at this beauty of the system. Now, these epithelial cells, which turn into the M cells or the membranous cell, they throw out their arms, which raises above the mucus, and then the bacteria actually slides and slips into this M cell. And once this slips into this M cell, they undergo a process of bacterial translocation to the underlying layer. Now, the underlying layer of the epithelial cell of these enterocytes are the lamina propria layer. And the lamina propria layer consists of macrophages, neutrophils, T cells, and the B cells, and every other cell which actually should give us protection. That means if a bacteria gets inside the neutrophils or the macrophages, they should be destroyed and they should be thrown out of the system. But pathogen being a pathogen and their subversion strategies are amazing, this does not happen. So the bacteria manages to get from our mouth into the small intestine, then inside the layer onto the macrophages and other dendritic cells. And there have been reports of few other mode of this entry. One of the very interesting mode is these dendritic cells. So the dendritic cells are professional antigen presenting cells, and they should be in fact capturing the pathogen and trying to process it for the presentation. So what these dendritic cells do, few of them, that they also throw up their dendrites. And these dendrites, they come out of these intestinal lining and they capture or sample the bacteria, and then they take it up. And once they take it up, instead of doing the process of presentation, now Salmonella hijack this dendritic cell for its dissemination. So macrophages and dendritic cells, they encapsulate or encapture the bacteria and they disseminate into the distal organ. And that no reason why Salmonella infection is such a notorious infection. It is just not now limited to the gastroenteritis or typhoid fever. You have 2,700 different serovars, out of which Salmonella typhi and paratyphi is strictly to the human, but all other can infect, from the cold-blooded animals to the avian species to the animals to the human. And it, according to the bioterror category, it is categorized as a class 2 bioterror organism due to the fact that we still do not have a very good vaccine candidate, and Salmonella is now known to cause extraintestinal infections like meningitis, endocarditis, and bone infections, etc. So it is indeed a matter of worry. Now, let us understand that why Salmonella is not able to when it is in the vacuole, why is it not able to come out into the cytosol? So first, let us understand that what do you mean by living in a vacuole? Whenever any bacteria, XYZ, any bacteria that you name, wants to get inside the cell, inside the host cell, there are different mechanisms like secretion system for induced uptake, other mechanisms by which the bacteria is going to be so kind of endocytosed or phagocytosed or encapsulated by the host plasma membrane. And this bacteria is now within the vacuole. Now, this you call it actually as a vacuole. That means there is a kind of a case around the bacteria. Now, the bacteria is within the vacuole, and this vacuole is known as a naive vesicle or the naive pathogen containing vacuole or bacteria containing vacuole. Suppose you put E. coli, simple DH5 alpha E. coli that you use for your molecular biology experiment, and you try to put up an experiment with E. coli, you will see that the moment the E. coli is taken up, within 10 to 15 minutes, it is going to immediately fuse with the terminal organelle, that is a lysosome. And lysosome are very toxic organelle which contains lot of degradative enzymes, the hydrolases, the cathepsines, etc., which is going to kill the bacteria right there and get rid of the bacteria. But what happens in case of the pathogen? Now that is the very interesting point. When the bacteria comes in this so-called naive vesicle, the bacterial containing vacuole undergoes so-called modification. Now this modification is from the naive pathogen containing vacuole to very mature pathogen containing vacuole. And there are a lot of very wonderful events that goes on where they share some of their proteins, like early endosome marker, EEA1, transferrin receptor, the RAB5, then the recycling endosome marker like RAB11 and RAB2, late endosome marker like LAMP1, LAMP2. These are all these several different organelle biogenesis pathway markers which comes and fuses with these Salmonella or the pathogen containing vacuole, and it makes it very mature. What happens in case of Shigella? Suppose this Shigella or Listeria, it goes by the similar pathway. These two bacteria are an excellent example of a pathogen in the cytosol, not in the vacuole, but in the cytosol. They cannot stay in the vacuole, so they don't like to live in a vacuole. And the reason is that they do not have the stringent machinery to survive within the vacuole. So the first thing what they do is they express their toxins, which is encoded by the EPA group of toxins or the Listeriolicin O, which is secreted by Listeria. And these two bacteria, they immediately lyse their vacuolar membrane with the help of this toxin, and now they are free in the cytosol. So their life is a cytosolic. So they're intracellular, but they become cytosolic in nature. They are not bound by a vacuole. They are very free. They have abundance of nutrition, but this abundance also comes with a cost. And what is the cost? Is that these two bacteria, they can be cleared very fast by the antimicrobial defense system, and also they can be very easily tackled by the help of the antibiotic, because they are free, kind of a free floating in the cytosol, and there is no way by which they can protect themselves. So, and also they come with one more very difficult kind of a process, that they have to move now from one end of the cell to other, and they need to use that host actin tail. So they polymerize the host actin tail and they move from one end of the cell to other. And then to cross from one cell to other, they have to shed their actin tail and again get into the other cell. So the process is very cumbersome and energy expensive, and that is the reason eradicated very fast. Now, when you come to Staphylococcus aureus or mycobacteria, these two pathogens, they try to arrest their phagosome, that is so-called the pathogen-containing vacuole, to one stage. One stage means that they will not allow this pathogen to mature and get immediately fused with the lysosome, because if they do this, then their life comes to a dead end. They modify it in such a way that they arrest it and stall it. You call it as a stalling process. You stall this pathogen-containing vacuole to the early endosome stage, so that they are very comfortable within this vacuole, and they will do all their necessary pathogenesis effectors, protein. They will secrete everything being in that vacuole, and the pH of this vacuole is not acidic, so which is very important for these two bacteria. So because they do not again have certain the two component system, which is going to give them protection against the acidic environment. Now let us come to Salmonella. Salmonella is extremely unique. It is not going to stall the maturation. In fact, the Salmonella will exchange factors with many of these organelles like early endosome or the recycling endosome. And also it harbors a marker of some of the late lysosome. And the pH of the Salmonella containing vacuole is acidic. It has a wonderful system to sustain in the acidic pH of the vacuole. So that is the reason that why the Salmonella is a very perfect and the strict example of a vacuolar pathogen, where we will now look into the strategies that how and why do they survive within the vacuole. So now, if you look into many different examples of pathogen which are there within the different cell type, say for example, this is the example of the Salmonella growing in the epithelial cells, and there are various reviews which have envisaged the fact that this is, say for example, a Salmonella which has entered through the M cell or through the process of enterocyte invasion and gets into the Salmonella containing vacuole. Now, the Salmonella also grows within the vacuole, and the earlier hypothesis before we stepped into this field was that the growth of the Salmonella, division of the Salmonella happens, and looks like that this Salmonella vesicle also grows. And so there are multiple Salmonella in a given vesicle, so that it's like a balloon. You can envisage it like a balloon, that 1 becoming 2, 2 becoming 4. So there's a logarithmic growth of the Salmonella, and then the vacuole is also kind of growing. So that was the picture that was envisaged before we stepped into the field. Now, when my lab began in 2004, I started with a lab with my very dynamic colleague of mine who was a medical doctor, Sandeep Ishwarappa, who is himself currently now a faculty in the Department of Biochemistry in Indian Institute of Science. So he, being a doctor, was a very interesting trait that he carried, and the trait was observation. It is always for him that the observation, similar way like he used to observe his patient, he observed the cells. He observed days after days, cells after cells, sections after sections. And one thing, when one night he came and told me, "Madam, I am seeing something very interesting. Whenever I stain a cell, say for example, this is an staining of an Salmonella infected cells. These are the macrophage cells, salmonella infected cells. And the green, what you see here is the salmonella, which is a GFP salmonella, salmonella expressing green fluorescent protein. And the RAP7 is a marker, one of the marker for SCV, salmonella containing vacuole. You have a many different markers for the salmonella containing vacuole, like LAMP1, RAP7, even cathepsines, etc. But LAMP1 and the RAP7, we take it as a marker for SCV. So he mentioned that whenever you see a salmonella infected in a cell, you always see them a single salmonella in a vesicle or in a vacuole. You never see a multiple salmonella in a given vesicle. So if you now see this particular micrograph, you will see that these vacuole is exactly stained in the shape of the salmonella. Salmonella has a very tight fitting vacuole, unlike Legionella, which has a very spacious phagosome. So this is an extremely tight fitting vacuole, and they indeed have, they reside in a single salmonella in a given vacuole. Now, this single salmonella per vacuole was very interesting to me and to him. So that time we were two with couple of project trainees there, and we wanted to, so we actually debated a lot. And every day it was a very exciting day for us because we wanted to see that is it an artifact or is it really true? So went on with cell after cell, animal experiments after animal experiments, and finally we figured out that this is indeed the case, and there is a very new novel mechanism by which a single salmonella stays in a single vacuole, and when the salmonella divides, the vacuole divides. Now, you will understand that the division of the salmonella is very well known. For that matter, all of you are graduate colleagues and many of you are studying microbiology. This is not very different to you from E. coli division, say in LV, that how the binary fission happens and how the symmetric division happens in a bacteria and how the growth curve is established. This is all very well known to you and to us. But when the bacteria is within the cell, and that too within the vacuole, how does it happen? The bacteria is dividing, I can understand, but then how does the vacuole divide? And the division of the vacuole is simultaneous and very well coordinated with the division of salmonella. How that can happen, and that became the focus of our lab, and everything now revolves around life in a vacuole. Life in a vacuole became our bread and butter. It became one of the most exciting topic all across the world now, and it also became extremely exciting to look into the mechanism. Confocal microscopy and any other methods of microscopy must be corroborated by the electron microscopy, because we must not forget that extrapolating any research of in vitro data or microscopic data should be always taken with a pinch of salt and with lot of care until and unless you really see those kind of changes or the processes happening in the electron, even under the electron microscopy. So now this is what our colleagues did in the lab, and they went ahead and they did an electron microscopic took the electron microscopic pictures on various type of cells which were infected, and even in the liver from the animal, to really see that this is not an artifact of only the cell, but it also happens in the animal. And thankfully, we are very lucky to have our mouse model. When mouse are infected with Salmonella typhimurium, it mimics a kind of a human typhoid fever. It is a dead-end model, and one can study a lot and know about the pathogenesis from the Salmonella typhimurium murine model. You call it as a murine model. So if you really look into this under the electron microscope, so you will see here, this is a single bacterium in a given vacuole. So indeed, you are seeing it as a round because the way by which you take a section, so the cross sections, you will see it in the form of a round bacterium. And the moment the bacteria, so my Sandeep and my other colleagues, they were very, I would say that they were very successful in catching a field where the bacteria is now dividing. Now very carefully look at this particular plane where the bacterium has already divided into two, and now your vacuolar membrane is also dividing. So it was like a kind of invagination, and now this is this two bacterium with its two vacuoles. So this black is the bacterium, and here what you see is the hollow is the vacuole around. You will see many photographs like this, where you see this bacteria which is enclosed in the vacuole, and whenever they are making their headway to divide, the bacteria is dividing, and along with that you see an invagination. Now, this kind of invagination of the vacuolar membrane was something which was extremely interesting to us. We talked with many of the mathematicians and physicists around us to understand that what is the force that the bacteria is generating, or is it something like another physical force that the vacuolar membrane is drawn inside. Now, this is one beauty of IISc, because you step out of your department and you meet chemists, physicists, mathematicians, and you can discuss your science with them, and they give you a lot of their ideas. And our physicist friend told me that no, this is not working out. The model is not working out by simply saying that it is a suction force which is simply pinching this particular vacuolar membrane in. There has to be something much more to the very active division of the vacuole along with the division of the bacteria. Now, what is it that is making this bacteria divide? So that became the next very important question. Now, if you really take a very closer look, take this extremely closer look here, and this is the section of the liver, Salmonella, which we infected the mouse and then took the liver sections. And this is the intestinal 407 cell line. So cell line comparison with the real liver tissue sections. And if you see here, indeed, the bacterium is in a single vacuole, bacterium in a single vacuole. Here also in the liver sections, the bacterium in a single vacuole. And this is the most convincing data to suggest that indeed the single bacterium per vacuole is just not being shown in the cell culture model, but it is indeed very true in the animal model. Liver is one of the very prime organ for Salmonella infection. The dissemination happens in the mesenteric lymph node, in the spleen, as well as in the liver. And in the liver also, you can see this is very beautifully getting surrounded by a vacuole and one bacterium per vacuole. And evidence that indeed when the bacteria is dividing, the vacuole is undergoing division. Extremely difficult set of experiment, and one need that keen observation and skill and a perseverance to in fact do these kind of studies, where sections after sections, more than 100 to 200 sections, need to be cut and seen under the microscope to indeed figure out that what indeed is going on, what processes indeed they are going on. So yes, we were very much convinced that it is a one bacterium per vacuole. The question is, why do they love so much their studio apartments? It is sometimes interesting to see that why studio apartments can be so lucrative. Instead of some of you maybe having a mansion and living there, and some of us actually, I still remember my studio apartment in Germany, that how comfortable it was versus a big mansion now if you compare. So why do bacteria love their studio apartments? There can be many reasons for their loving it. First is, they definitely have a very reduced competition for the nutrient, because imagine having a very big room with so many people around and one source of food. So everything is kind of distributed and the competition will be much more. So the studio apartment, one-in-one, have a much less competition for the nutrients. They also have an increased surface area for nutrient acquisition, and they have also enhanced surface area for each of the bacterium to secrete their effector proteins into the host cell milieu. Because the effector proteins, which are secreted by the bacteria, are one of the prime proteins, or we call it as a virulence protein, which will in turn decide the fate of the host cells. So if the single bacterium in a vacuole, each of them can secrete, the strength of the effector proteins will go very high, in turn establishing the pathogenesis in a very wonderful way. And last but not the least, the host cell must strategize to attack the individual vacuoles. If the number of the vacuoles, bacteria-containing vacuoles are more, and if you have very few number of vacuoles like lysosome, the toxic vacuoles like lysosome or organelles like lysosome, then you can imagine that the single lysosome could have fused with bacteria, vacuole containing many bacteria, and that would have in fact killed everything. But in this case, it will not happen. What is the reason it will not happen? Because they are all packaged in a single vacuole. So maybe the lysosome number may not be enough to kill all of them. So maybe that is the reason that they love to live in their very stringent acidic vacuole, which is not having enough nutrient. There is absolutely no kind of, the luxury that they would get, but they become an extremely successful pathogens in that term. The question is, how do this studio apartment or their vacuole divide? The idea of this division came from, again, Sandeep and couple of my other colleagues who mentioned, hello? Okay, no questions. So they came to one very interesting observation, and the one interesting observation and the key factor that they derived from one of the cell's own organelle, that is the mitochondria. Now, all of us know that mitochondria is none other than alpha proteobacteria. It is the best example of the endosymbiont in the course of evolution of the mammalian cell. How beautifully this alpha proteobacteria, in fact, harbored the cell, got inside the cell and became one of the very important organelle. And not only that, it became one of the very important organelle, similar to that of plastids or the chloroplast in the plant. Mitochondria also have its own way of mitochondrial division or the mitochondrial fission. And it carries its own gene, its own genetic element. And there is a lot of crosstalk between the mitochondria and the cell nuclear proteins and the gene expression. But the mitochondria is an entity where we can derive our entire idea from, that why the vacuole and how the vacuole divide. Now, mitochondria has two membranes, that is the inner membrane and the outer membrane. Now, the inner membrane division, this particular inner membrane, which you are seeing it as a folded cristae, this inner membrane division protein has a homologue of FTSZ, which is termed as a MGM1. Now, FTSZ is a very important and one of the most crucial protein in the cell division mechanism machinery in the bacteria. Now, FTSZ is an homologue of tubulin, cytoskeletal element, and if bacteria is having any perturbation in the FTSZ, then it becomes a lethal mutant. So FTSZ is the most important protein in the bacterial division and also in the maintaining the architecture of the bacteria. Outer membrane actually contains these two very important proteins, that is a dynamin related protein 1, DRP1, and HFIS1, which is important for the outer membrane division. So the inner membrane and the outer membrane division protein, they coordinate in a very wonderful way, and then the mitochondria divides. So this is how it divides. This is in a schematic that the DRP1, it marks the site of the division, and the source of this energy is the GTP to GDP and PI. And then finally, the one mitochondria divides into two. Now, having said that, is it actually some kind of, idea that we can draw from the mitochondrial divisionary machinery into our Salmonella containing vacuole division machinery? Now, to begin with, let us mark it out. Let us mark it out that what happens. Now, if you see here that this is the cell which is infected with the Salmonella, and this is this M-Cherry marked construct, M-Cherry KDEL construct, which is going to now mark out your site for the bacterial or the vacuolar division. And now if you indeed see this, so the M-Cherry is expressed all over. And when you do your infection, now you look here, this is this green Salmonella. And when you see this invagination of these M-Cherry KDL sequence, that it is indeed forming a kind of a marking or a kind of an invagination towards the site of the division machinery. Now, this work was done by my colleague Dabolina. So Dabolina followed Sandeep's work to indeed figure out that what is the molecular mechanism or what is the machinery that marks the site of the vacuolar division. So if you see this, this is the bacteria. Surrounding it is this SCV, and this is wrapped by endoplasmic reticulum. So what she figured out was that that actually the site of the marking is really very dynamic. It is brought about by wrapping of this endoplasmic reticulum around the so-called SCV, thereby recruiting certain proteins like dynein-related proteins 1, dynamine-related proteins 1, etc., to mark the site of the division, and finally the vacuolar division happens. We are trying to show it much more by the live imaging microscopy to really understand this molecular mechanism of vacuolar division. So dynein and salmonella have a relationship in terms of a transport system. Say, for example, it was reported in Molecular Biology of Cell and Journal of Cell Science that the dynein-mediated vesicle transport, it controls this, the division of the salmonella in the in vitro situation in the cell line. And the microtubule motor, it controls the membrane dynamics of the salmonella containing vacuole. So now let us understand this complex. You don't have to worry about this complex at all. It is just to tell you that sodium orthovanadate is a chemical which inhibits this dynein. So if you try to inhibit the dynein, so Debolina asked a question and even Sandeep asked the question that if you inhibit the dynein, does the replication of the salmonella and the salmonella containing vacuole comes to a halt? This is the first question they asked. And the second very interesting tool that we have is P50 dynamethene. Now, P50 dynamethene negates the action of the dynein. That means if you have a large amount of P50 dynamethene in the cell, it is going to inhibit the function of the dynein. So we have this construct of P50 dynamethene as well as the sodium orthovanadate, and then asked a question that what is the situation now? If you look from this data, even in the cell science, that if the salmonella is treated with the P50 dynamethene expressing construct, wherever you have an overexpression of the P50 dynamethene, then the average number of the bacteria goes down. But they did not know that why it is happening or what is the reason. They just gave an observation that it goes down, and they stopped there. And then finally, when we indeed looked into the action of the orthovanadate or of the P50 dynamethene, we had hit upon something very interesting. Now, if you see here, look at the control panel. Now here the green is a salmonella again, and red is one of the SCV marker, and the marker is none other than the LAMP1. It is a lysosomal associated membrane glycoprotein, LAMP1, which is a characteristic marker of the salmonella containing vacuole. So the control cells which are untreated, you will see here this wonderful single bacterium per vacuole, all single bacterium per vacuole. But the moment you treat them with sodium orthovanadate, 100 micromolar, which specifically inhibits the dynein, though sodium orthovanadate has many, many different targets, but this is the concentration which was known to inhibit the function of the dynein. You immediately see that the bacteria is coming in a single vacuole. Now, all of these bacteria are multiplying, but the vacuole is not multiplying. So amazing. Bacterial division is not halted, but the vacuolar division is halted. So it has come into the single vacuole. So we call this particular stage as a vacuole containing many salmonella, or the VCMS. And these VCMS were counted. Now, what is the fate? Okay, the bacterial division is not hampered, and you indeed see that kind of a growing balloon filled with salmonella. But what happens to the fold replication? Now, if you see the percentage of the cells which are showing this VCMS phenotype, the cells when they are treated with sodium orthovanadate, you see a very high percentage of the cells with these kind of VCMS phenotype. But finally, what happens to their intracellular growth? You will see that there is a drastic reduction in the fold replication of the intracellular growth from more than 10 to 15 fold to less than 2 fold. That means though they are able to grow or replicate to some extent, but if the vacuolar division is hampered, there is no way that the salmonella will carry on with their life and establish the pathogenesis. And as a whole, the entire population falls. And because these bulky, so-called bulky vacuoles are in fact targeted by many of the antimicrobial peptide containing vacuoles or lysozymes, and they all meet with their dead end. So they are killed. Their effect is gone. Now, once this particular data was obtained, our joys had no boundary, because we thought that we had hit upon something very, very important. So look here. This is the cells which are expressing this P50 dynamethene. So if you have the cells which are expressing this P50 dynamethene, which negates the function of the dynein, you see the same effect of many VCMS here, and their number also comes down. So which shows that the dynein motor protein is one of the important motor protein to exert the to start the vacuolar division, exactly coinciding with the bacterial division. So this is indeed very important for the bacteria to divide its vacuole, and if it is not dividing the vacuole, it is totally getting perturbed in its intracellular life. Having said that, the one very important question came to our mind, and we had hit upon something very interesting, is that can we now think of an intervention strategy, not against the bacteria, but against the division of the vacuole itself? If you stop the vacuolar division, you're going to automatically stop the bacterial growth and the spread of pathogenesis. And the small molecule inhibitors and etc., it is in progress to figure out that how we can stop these interventions. We are collaborating with many chemists across IISc and trying to understand and screening some libraries to figure out that how we can stop this vacuolar division. Yes, it is very true that first experiment you do is try to figure out that whether your DRP1, that is a dynein related protein 1, indeed co-localizes with Salmonella, and it does co-localize with the Salmonella. This is the electron microscopy pictures of the DRP1. This big blob that you're seeing is the immunoelectron microscopy to tell that the DRP1 indeed are juxtaposed or they co-localize with the Salmonella. And maybe this is the one which is inducing, just like in the mitochondria, the mechanism of the vacuolar division. Now, what if you express the dominant negative DRP1? Now, you have to definitely prove beyond doubt that DRP1 is the protein of interest, which marks the division site, and it is important for the division. So when you express this dominant negative form of the DRP1 into the cell and ask a similar question that whether now it is going to give you the division, the answer is that the moment you negate this DRP1, then you will again see that their entire effect is lost, and then they come as a multiple bacteria in a given vacuole. So the wild type DRP1 or the intact DRP1 is extremely important for the bacteria to undergo vacuolar division along with the bacterial division. Now, from the Salmonella typhimurium virulence factor, we have our lab is our expertise, and our colleagues' expertise in the lab, it lies with generating a mutations or the mutant or the knockout strains in the Salmonella typhimurium. So we have a battery of the mutant in the lab. All our colleagues and now Akshay, they have generated a huge number of the mutants, which are of very much importance to understand the basic biology of each one of the protein in Salmonella. We have it in our stock. We also make a GFP and the RFP constructs so that we can visualize them very beautifully. So one of these mutant RFAG, from our stock, it showed that if Salmonella is devoid of this RFAG, particular gene RFAG, then again you get this phenotype of a multiple bacterium in a given vacuole. Now it looks that this is a very, an effort, extremely coherent effort from both the host side as well as from the bacterial side to make this one bacterium per vacuole. If the host does not have the dynein, intact dynein, and it cannot be recruited to the site of division, then also there is an issue with the vacuolar division. If the Salmonella do not have this RFA gene, then also there is an issue of the vacuolar division. So both of these proteins interplay with each other from the host and from the bacterial side to give you the single bacterium per vacuole. And if you do not have this RFAG, again you see these confocal microscopic pictures which shows that there are multiple Salmonella that are coming in a given vacuole. So yes, we have hit upon many of these targets, and we now know that what are the important players leading to the single bacterium per vacuole. Now, divide and rule is the key here. For Salmonella, it is not in the unity, it is divide and rule. If the Salmonella divides with its own vesicle, then it is going to stay beautifully. The same in macrophages or in the epithelial cells, any kind of cells that you take, it is always a divide and rule. Look at these beautiful epithelial cells which are filled with salmonella, all in single vesicle. All these round structures that you are seeing, these are nothing but the green bacteria with the red for the lamp staining. So each one of them, they are in the intracellular, intravacuolar bacteria living in a vacuole. So they all have their hut, that is the single bacterium per vacuole is the key here in any cell type that you take. And if you now, as I mentioned to you, that if you now start scoring for the cells, whether they are in a single bacterium in a vacuole or a multiple bacterium in a single vacuole, you will see that these particular mutant that I mentioned to you before, RFAG, the moment you delete this particular gene, you will see that it will be a multiple bacterium that is coming in a single vacuole. So this is M83 is another coded mutant, just to show that this is a very specific mutant which leads to the multiple bacteria in a given vacuole, whereas the wild type, it's just the opposite way. It is a single bacterium in a given vacuole. So extremely convincing data to suggest that these particular bacterial protein RFAG is very important for the bacteria to get into a single bacterium per vacuole. Now, the dynamine related protein 1, when you want to really look into much more detail, it is, I'm just showing you because, you know, it will also allow you to think much beyond what is in the cell. Whenever you are working with a bacteria, first of all, it is very difficult to work with bacteria in vitro. It is not as easy as we think now. We always thought that bacteria are kind of, they show this kind of an immortality, immortal. You can take a scoop and you can put it in the tube and it will grow and everything is okay. But that is actually not the case. Now let us start thinking that the multicellularity, the concept of the quorum, the concept of multicellularity in bacteria needs to be appreciated. And we also have to appreciate the fact that bacteria works in a community. The communal behavior is the most important key. Having mentioned about the communal behavior, what is the communal behavior in the cell? Now, in the cell, where the bacteria are separated by a vacuole around them, how do they crosstalk with each other? This is our next very interesting question to boil on, that what is the crosstalk or what is the quorum signal that goes on within the in vivo situation? What one can do is one can in fact isolate this vacuolar bacteria from the cell. So you isolate this salmonella containing vacuole. We have a very good technique to do that. You lyse the cell and with certain step you isolate them. So now you don't need the cell. You have the salmonella which is in the salmonella containing vacuole. You see this is the simple confocal imaging and this is the bright field imaging of the salmonella, green salmonella encapsulated or enclosed in the vesicle. And when you do your immunogold electron microscopy, you indeed see in isolation that these DRP1 or the dynamine related protein 1 does get localized with the salmonella containing vacuole, showing that there is a recruitment of the DRP1 to the salmonella containing vacuole. Now, how are they recruited? What are the factors that make them to recruit and how do they travel? See, DRP1 is a very important protein for many functions, including the mitochondrial division. So how the bacteria manages to hijack this DRP1 complex and then do this job. So this is all a very interesting, field of research that is going on. And the more you think, the more you will get fascinated. I will, kind of leave you here on this topic with this life in the vacuole. And we will definitely talk during the question answer session. But now let me take you to something very interesting that also is a kind of a very important, important molecule to do with the vacuole. It is not the bacteria in the vacuole, but the cells of the vacuole which are toxic. Now, all of us know that cells like macrophages, they are loaded with lot of these granules or the vacuoles which are important to take care of the cell whenever there is any invading life form, invading pathogen or bacteria or virus. It can be oxygen independent, oxygen dependent. There are many of these vacuoles have antimicrobial peptides in them, like lactoferrin and the lysozymes. Lysosome itself is a very wonderful organelle, etc., etc. It was very interesting to see that in the human macrophages, there is one very important protein that is not in human macrophages, in general, in the macrophages, there is a very important protein which is known as a BPI, that is a bactericidal permeability increasing protein. Now, as all of you, as a graduate student in microbiology, it will be very much interest to you to learn a bit more about that BPI molecule. Now, as the name suggests, it is a bactericidal permeability increasing protein. What does it mean? Means this particular protein is very, very important when a bacteria attacks the host, because this protein, in fact, it interacts with the lipopolysaccharide of these gram-negative bacteria, and they perturb their bacterial function, and they also takes a big toll on the bacterial life. So this is indeed one of the most interesting protein that is there, that is present in the human macrophages. But unfortunately, it is not found in the mouse macrophages. Now, all these years, always there is a correlation drawn or extrapolation drawn from the raw macrophages, from the murine macrophages to the human macrophages. But this was kind of an exception among many, which showed that if you look into this particular level, the transcript level of the BPI, this particular transcript or the gene is present in the, is expressed in the U93 cell, which is a human monocytic cell line, but is not found in the raw macrophages, which is the mouse cell line, mouse macrophage monocytic like cell line. So this is the cDNA prepared from the human PBMCs, and you see a beautiful 1.5 kb transcript, but that is not there in the mouse. Is that the reason that a lot of the research which was done before by the BPI in fact failed, because they did not know that raw macrophages in fact do not have this BPI? So further human PBMCs, when they were stained with antibody to the BPI, you can see that it beautifully stains red. That is, the BPI is spread all over in a granule, and CD11B is a marker for the PBMCs. So indeed they are present, and they are present in a kind of a granule. Now, is the characteristic role of the BPI, that is to take care of the bacteria, is that indeed true? So this work was done by my colleague Arjun Balakrishnan, who is currently doing his postdoc in Umeå University in Sweden, Stockholm. Now he wanted to understand that what is this expression level of the BPI in the human macrophages, monocyte macrophages, and he figured out that all of these particular PAM, say for example flagellin or the whole bacteria, Salmonella typhimurium or Salmonella typhi, etc., they in fact upregulated or induced the expression of the BPI in the PBMC population, showing that indeed there is a loop, there is a transcriptional upregulation of the BPI whenever you put any kind of the bacterial PAM, like flagellin or the lipopolysaccharide or the whole bacteria. And even in the macrophages, if you see here that in the macrophages, the Salmonella typhimurium also induced the upregulation, the fold induction of this BPI. But when you have the delta fly C, now STM, Salmonella typhimurium, one of the very important PAM or the associated pattern is a fly C, which is none other than flagellin. Now, flagellin or the flagella is the PAM for TLR5. So the moment there is no fly C, the expression of the BPI went down, which indicated that it is very much possible that this particular fly C is one of the PAM for the expression of the BPI, which also you can see that if you put a purified flagellin, it also induced the upregulation, showing that this may be one of the PAM for the expression of the BPI molecule. And then when you do this, confocal imaging or the staining with an antibody to BPI, you indeed see that there is a very beautiful staining pattern of the BPI when the bacteria, when the cells are treated with flagellin, or even when you treat them or infect them with the whole Salmonella. Now, what is the next question that comes to the mind? If there is so much of BPI that is being made, is the bacteria really killed by the BPI in the system? So the next experiment that Arjun did was, he first wanted to look at what actually that BPI does. And in a nutshell, what he figured out was that BPI in fact mediates the bacterial entry in the human macrophages. If the BPI was somehow downregulated with the help of having some kind of a construct, double-stranded RNA construct, which will inhibit the expression of the BPI, then the cells, the Salmonella typhimurium, was not able to get inside the the PBMCs or the U93 macrophage cell line. Look at the agony here. The BPI, which is expressed on the surface of the U93 macrophages or human PBMCs, they were supposed to actually take care of the bacteria then and there. But the bacteria is using it as a very wonderful opsonizing, kind of an opsonizing agent to get inside the macrophages. So this strategy, under any circumstances, to make something so, amenable to their own life is remarkable in Salmonella and of course, in many other pathogens. But I'm so fascinated as a student of microbiology, and I will always remain a student of microbiology. This particular field is teaching me a lot. And I'm not only learning every day, but if I myself put in the shoes of a pathogen, I'm really boggled by the way they think. Now, this is an example that it is indeed true that it is taking the help of this particular BPI, which is a toxic molecule, to get inside. And if you really do not have BPI, then the bacteria cannot get inside. So this was, in fact, the first finding of its kind to understand that BPI mediates the entry of the bacteria in the human macrophages. And that is not true with the gram-positive bacteria, Staphylococcus aureus, where in the presence or in the absence of the BPI, both these cells can take up the Staphylococcus aureus similarly. So this is indeed an extremely interesting finding. But with this finding, the first thing that came to the mind was, what if the BPI now is allowing the bacteria to enter, and that BPI should have actually killed the bacteria in a due course of time? Then if that is so, then why that is not happening, and why the bacteria is becoming, is proliferating in the given cell? And now if you indeed look into the system, that the STM actually shows less BPI interaction in macrophages during their replicative phase. What does it mean? The bacteria, say, only follow this particular panel. It is at 15 minutes, it is seeing the BPI, it is taking the help of the BPI, and it is getting inside the macrophages. Now 30 minutes, this attachment site, and within 1 hour, they are inside the macrophages. Now, once they are inside the macrophages, in fact, they come, like we talked about the naive vacuole, they in fact come along with the BPI. So what happens to the BPI? Arjun in fact found out that this BPI somehow over a period of time become less and less and less. How the BPI is degraded once it is, decorating the bacteria, where the BPI is vanishing. And if it is vanishing, why is it vanishing? It is definitely vanishing because otherwise BPI would have killed the bacteria, which is not happening. Now, if that is not the case, where is the BPI vanishing? And this vanishing of the BPI, or the less and less staining of the BPI, in fact led to something more interesting, that the degradation of the BPI in the actively recruited Salmonella containing vacuole is a very different procedure, by which the bacteria want to keep something which is very important for them, but will simply get rid of it by recruiting other proteasomal degradation machinery to get rid of this molecule like it is a BPI. Now, to get that example that it is indeed true that the vacuolar bacteria gets inside with the help of the BPI, then degrade the BPI so that it can stay stable. Arjun took an example, took an example of a Shigella flexneri. So he put up an experiment with Shigella flexneri because Shigella is a purely cytosolic pathogen. So being a purely cytosolic pathogen, he wanted to ask a question that what happens in case of Shigella. So if you see here that if the bacteria, if the cell does not have BPI, then the flexneri survival goes very high. That bacteria maintains a very active replication like cytosolic bacteria only when you do not have BPI. And that may be now the reason. And because of this particular experiment, we now have got a reason that why Shigella is not a very successful pathogen. Because the BPI induction in the cell, in fact, actually kills the Shigella. And if you cytosolic Shigella, which it cannot do when the bacteria is in its own apartment, that is in the vacuole. But if the cells do not have a BPI, if you downregulate the BPI, the Shigella are very, very successful in growing and they maintain their niche. So indeed, it is very true that the antimicrobial peptide, though it can play a very important role in the defense system, how the pathogen, try to get it out of them as and when required. Again, a vacuolar to vacuolar fusion. And whenever the bacteria is not in the vacuole, this is one example that how the life in the vacuole is very interesting and it is very safe, because if it is not in the vacuole, then it can get very readily attacked by the antimicrobial peptide. Let me switch gear and tell you the story of a totally different field, that is from human, mouse to plant. So when the lab was very actively working on the role of Salmonella and the vacuole and etc. in the system of animal, using animal model and from the, the human cell lines, etc. Kapudeep in my lab, he joined from the agriculture background. He's an agriculture scientist. He's currently faculty in the Uttar Banga Krishi Vishwavidyalaya. And he came up with a plant system as a model. And during that time when he joined the lab, there were many reports of the recurrent multi-state outbreak of the Salmonella, Newport, which was associated with tomatoes. Oh, we had really no clue that Salmonella will even not spare plant. Okay, it has not spared anybody, including the snakes and including the birds and including crocodiles, reptiles. But we could never imagine that it will not spare plant. Now, when these reports came out, we understood that we thought, actually it was given in the report, that all these different kind of outbreaks that one is seeing is because of the contaminated irrigation water or a post-harvesting handling process. So the entire California farm, it was, stopped, and there was a huge economic loss on the tomatoes, because the tomato products and the raw tomato as a whole was looked upon with really yellow eyes. And what is the reason? When Kapudeep joined, he actually figured out that it is not a post-harvest problem. Salmonella enters the plant through the roots and none other than the lateral roots. Now, many of you who are in the plant field and in the plant science, you will understand the emergence of the lateral roots. Now, there is something known as a remodeling of the epidermis when the lateral root emerging is going on, and it is a very small natural wound. Beautiful, ripe looking tomatoes will be very nice to look. You see here how the Salmonella is traveling. through these and it becomes systemic in plant, and it looks very beautiful. It is red in color, and one may eat it and then get and suffer from salmonella-induced gastroenteritis, or most probably gastroenteritis, because we are still to do work with Salmonella typhi and whether it becomes systemic. Now the contamination of the salmonella in the also have been used to understand that remember so of raw fruits, the raw salad, how safe they are, because the salmonellosis by these salad vegetables, they are spreading now. And
Not audible and not visible.
Good afternoon. There is a small technical issue with the network. We'll get back to you immediately.
Okay.
that I told.
Deepika ma'am, you are there.
Can you hear me now? Hello, can you hear me?
Yes,
Yeah,
we can
ma'am.
hear you.
Yeah, you are audible.
Yes.
Thank you. And I was about to close my session, and my salmonella took me back to my hut. Anyway, in fact, I have finished my talk, but let me just share the slide to show you my wonderful group. And it is indeed true that you know how. Okay, let me quickly share the slide. Hope you can see my slide.
Yes, we can see.
Yeah. So I was about to just end my talk by telling that it is indeed very fascinating to see that you know how the salmonella can create a havoc, and how the host, different kind of the host, they are playing a very important role. So we actually established the so-called transkingdom cycle. I have not, you know, told the work of Akshay, who works on the delivery of the, you know, the antibiotics and the vaccines through the shock, which is a very big study in the lab to understand the wound healing and the other process. So now what I would do is I would very quickly try to understand, make you understand certain avenues in IISc that one can come through. So this is a kind of an advertisement that I'm doing for my own institute, because I know that I have students from all across India, and all my very eligible teachers and wonderful teachers who train the students. And thank you to all of you to producing such amazing students. So you can get into the institute for integrated PhD program through the JAM. All of you know that, and all of you, I'm sure, must write the JAM exam. For the PhD, kindly, you know, come through the CSIR GATE, DBT, or Inspire. Once you clear these exams, you're called for the interview, and then you join our lab, you join IISc, and you join any department that you may like to join, and then contribute to this exciting research that is going on. It is also very important to see that these are the only 5 characteristic features that you may have for you to really do your science. That is the eagerness, the vibrant nature, your ability to ask questions, your curiosity, and your persistence. And the why and how. This why and how is very important for you to ask. And these 4 or 5 criteria is required. And this is my wonderful group. All my colleagues who are in yellow here, they have all graduated, and they are faculties elsewhere across India, across world. These are my current PhD colleagues now with me, and I'm very thankful to all my granting agencies for the grant that they have provided to do the research. And indeed, it is true that there is something incredible that is waiting for us to see. I would like to thank IISc. I would like to thank Hislop College, my dear friends, my dearest friend Sunita, for giving me this wonderful opportunity, and all my amazing colleagues. I cannot thank you enough, because it is not due. Nothing can be possible. My lab also cannot stand if you wonderful colleagues, all who are listening, will not come to the lab. So thank you very much. And now we will open up to question. Thanks a lot.
Thank you, ma'am, for the enlightening talk. I'm sure we are all motivated to pursue a career in research. I now invite Ishek Shah, an MSc Biotechnology student, to take over for the Q&A session.
Thank you, Srishti. Let us begin our Q&A session now. Before we start, I would like to give you all a brief idea about how we are going to conduct the Q&A. To avoid overlapping and interruptions, I request our audience members to put in the questions that they have for our speaker in the meeting chat box or in the WhatsApp group that they're in. Our viewers from the YouTube live stream can also put in their questions in the comment box. I will read out the questions as they come in, and they can be addressed by our guest speaker. So moving on to the questions now. The first question that I have for ma'am is that do Salmonella never live in the cytosol? Ma'am?
Yeah. Okay. So may I know who asked the question so that I can actually address?
Ma'am, it's from Karen Thomas, a student.
Okay. Yeah. Thank you, Karen. Thank you for asking actually a very important question. Now, these questions of yours will in fact bring you to one of the very important concept, and the line which is drawn between a cytosolic and a vacuolar pathogen. Now, if I tell that the Salmonella is strictly a vacuolar pathogen, that means that they never quits the vacuole. And your question is it really true? And do they never live in the cytosol? The question is very valid. And why is it very valid? Because in fact, there are kind of maybe 1 to 2% of Salmonella, which is seen in the cytosol in epithelial cells, but never ever in macrophages or in dendritic cells. When you make a mutation in a gene which makes the Salmonella come out into the vacuole, that means a gene which cannot maintain the vacuole, those bacteria, they can survive to some extent and can survive to epithelial cells, but not in the macrophages. And the reason is that the environment of epithelial cells is much milder than that of the macrophages or dendritic cells. Macrophages are loaded with reactive oxygen species, nitrogen species, whereas epithelial cells are not. They are not a good producers of these two rapid oxidative bursts. And also, one very important thing is, if the bacteria is in a cytosol, then the vacuolar fusion, the fusion between the oxidative burst containing vacuole, that is which is guided by these, the GP91 FOX, and the Salmonella containing vacuole cannot happen. So this dynamic fusion cannot happen, and that is why the bacteria is much better in case of epithelial cell, and they survive. So yes, you do indeed see a small percentage, but it is not the life of Salmonella, and almost more than 99% of bacteria will be in the vacuole.
Thank you, ma'am.
Thank you.
So we have another question from Nidhi Patil, who asks, since vacuole is acidic in pH and the cytosol is around neutral, How do the proteins secreted by the salmonella survive in such a drastic pH change? Do microvesicles play a role in it?
Okay. If I am not mistaken, is it the same Nidhi who did a project with me and now is IIT Mumbai?
Yes,
She
ma'am.
was a student.
Yes, ma'am.
One very important. And SRA, SSRB, which then switches on a type 3 secretion system. And because of the secretion and making of the type 3 secretion system apparatus, Salmonella directly secretes those effector proteins into the host cytosol. Yeah. And once it is inside the host cytosol, then they do a different function to perturb the cellular other processes like inhibition of the antigen presentation or inhibition of the fusion of the toxic vacuoles with the salmonella containing vacuoles, etc. So if the vacuolar pH is made alkaline, or if the acidity is neutralized with the help of, say, for example, bafilomycin A, which is an inhibitor for VATPase, then the SPY2 system will not be activated and salmonella will die. So they like their acidic niche because that niche is a trigger for them to secrete the virulence factors.
Okay. Ma'am, also one more question.
Yes.
Ma'am, since salmonella is affecting plants also, so is it harmful to the plant or no?
Yeah. Nidhi again?
Yes, ma'am.
Yeah. Yes, Nidhi. So is it harmful to the plants? See, what happens is in the lab, what we have seen that if you have a very high burden of salmonella, the plant undergoes something like a process of chlorosis. It droops. So this is a kind of a dead end, which we did it in Arabidopsis in the model system. We will do a lot of genetic intervention studies now. But when you talk about tomato, the real field tomato, then in those plants, if your plant is infected, I mean the soil is infected, the tomato becomes much smaller and their size decreases. So there is a big loss. Even there is a loss. Though there is no difference, when you look at the tomato, they may be just smaller in size because they are filled with salmonella. But yes, if one consumes them, then it leads to the further course of pathogenesis in anybody those who have consumed.
Thank you, ma'am.
Yeah.
Thank you, ma'am.
Thank you.
Another student from our live stream asks, Chaitanya Gadekar, his name is. He asks, what do you think are the responsibilities of common people to contribute to reduce the emerging problem of antibiotic resistance?
Okay. So Chaitanya, yes, common people's responsibility is in fact huge. Now, sometime I would request Sunita to have a very informal talk about antibiotic resistance because now we are working a lot on this. And also we are understanding one very important point. That what is a journey of a bacteria or of a pathogen towards getting towards its development of the antibiotic resistance or antimicrobial resistance? Is it indeed the induced mutation or is it the selection? And how is the fate written? And why do they do not revert by creating another mutation which will now make them susceptible? Now, as a common man, my and your responsibility is huge, which is just one very important point. The first thing is that, as all of you also know, and your teachers also might have told you, that never self-medicate, and use of over-the-counter antibiotics should definitely be prohibited. And one very important thing, always take care of your gut flora. Because remember, if your gut flora, which is made up of 230,000 different species, and you should keep it safe because that flora itself now tomorrow should not become antibiotic resistance. And if it does become, it may spread it to the other pathogen which comes in. So without prescription, the antibiotic consumption should be totally prohibited. And doctor's prescription is definitely a must. So we should not be doing self-medication. But is it really only limited to self-medication? The answer is no. Where are all the effluents going from the antibiotic industry? What is the level of antibiotic in the water bodies? What is the level of antibiotics everywhere? in the food products, in the packet products that you're eating, in the poultry, which is, known to be now a very major source of the antibiotic resistant spread. Where are they? Can we do now something about it? It is a very active effort. It is an active effort of you, me, and you're spreading it across the entire generation, that how we need to be extremely vigilant. So it is really an active effort to give talks everywhere, including the industries, to figure out that what are the policy changes, what is the disposal policies, and is there any body which actually takes care of the total antibiotic burden in this universe? Who are they? And what is the control and the strategic measures that anybody would take to address this question?
Thank you, ma'am.
Yes.
So from our live stream again, Shibin asks, what are the major contents of a vacuole? Is it polypeptide or polysaccharides? And how does the salmonella get energy from it? What are the sources of energy for the salmonella? And are there transporters for the same?
Great. So salmonella is enclosed in we call it as a salmonella containing vacuole because that membrane contains salmonella. Now, what is that membrane made up of? It is first of all made up of lot of host proteins. So there are more than 40 to 50 odd host proteins which are recruited to make that salmonella containing vacuole. So right from this lysosomal associated membrane protein to the tubulin to actin to many of the transporters like cationic amino acid transporters, glucose transporters, and cytoskeletal element. Some of the very interesting component are antigen presentation machinery, MHC class, and we do not know that why it should be getting targeted to the SCV, maybe because SCV can be a very good organelle for the cross presentation. So many host protein, more than 40 odd host protein, and some of the bacterial protein are the one which makes the so-called salmonella containing vacuole. So in fact, we are going to do a very detailed study of the analysis of the salmonella containing vacuole now with the help of mass spec, for which you have to isolate the SCVs and then do for the complete proteomic analysis and so-called metabolomic analysis to understand that what all decorates. It is such an interesting interface between the host cytosol and the bacteria. And you will not imagine how clever this bacteria is. What is the reason for salmonella to recruit cationic amino acid transporter to the vacuolar membrane just to transport, say, cationic amino acid? Example is L-arginine. It was shown in our own lab. Bacteria can synthesize L-arginine. It has all the genes, but it requires 4 ATP. But if it just upregulates its transporter, it only requires 2 ATP. So whenever there is abundance of this L-arginine, it never turns on its synthesis gene. It will only turn on its uptake gene, and it also recruits the cationic amino acid transporter to the host, to the vacuole. And from the cytosol, it enters the vacuolar environment through this MCAT transporter. From the vacuolar environment, it uses the ARCH-T transporter of the bacteria to take that L-arginine into the bacteria. So look at the strategy. It's not going to spend a single energy, single ATP, if it can quench from everywhere. Within the bacteria, its entire metabolic system is very much intact. So it does like that. So it upregulates some of the transporters so that the metabolites and the essential amino acids can be taken up. And it continues to grow, to divide, and there is a certain threshold. So also there are, a lot of very emerging, amazing field to understand what is the threshold of the bacteria, salmonella containing vacuole in a given cell. How is the quorum established within the given cell when the bacteria is next to each other, but they are separated by a boundary? How that happens? So there are many burning questions which are waiting to be answered. Okay, so there is a question by Abhinav. Why salmonella choose only vacuole and not any other organelle or microsome? Good question, Abhinav. See, remember, whenever the bacteria is inducing its uptake or getting phagocytosis, what is the first vesicle or what is the first membrane that encapsulates the bacteria? That is the cell's own plasma membrane. So it actually comes with that naive vacuole around, and then it does not stay naive anymore. It wants to acquire more and more, and it only acquires more and more of the good things. On one hand, you have that organelle biogenesis going on from the early endosome to the late endosome to the terminal lysosome. This is one end is going on. And other hand, you have the Salmonella, which is using those same proteins which are used by the organelle biogenesis synthesis towards making more and more of the Salmonella containing vacuole. So it is actually making it. And they come by default, they come with that plasma membrane around them. And from there on, they go on modifying it by getting everything, all the goodies to their vacuole.
Okay, ma'am. Thank you so much.
Thank you. Abhinav, where are you from?
I am from Nagpur only.
Very good. What are you doing?
I have just finished my BSc just given my final semester exams from the Nagpur University, and I have I have also cleared some of the entrances of the central universities. So I am just applying for MSc now.
Excellent. Please come for PhD to ISC, to our lab. Yeah.
Yes.
All the best, Abhinav. Yeah.
Yes, ma'am. Thank you so much.
Good. Yeah. Oh, okay. Anisha Philip is a question. What is the kind of interaction between DRP1 and vacuole? Non-covalent or covalent protein-protein interaction? A very good question. And also not really known. But let me tell you one very important point, that the DRP1 is recruited to the Salmonella containing vacuole. And the DRP1 also, in fact, gets colocalized with the FIS1. If you remember that FIS1 was one of the protein which was responsible for the outer division of the mitochondrial membrane, the outer mitochondrial membrane. Now, the FIS1 and the DRP1 complex, that entire complex is kind of, hijacked towards the invagination, that is making the division in the Salmonella containing vacuole, that is dividing the Salmonella containing vacuole. So what is the protein in the vacuole with which it is interacting, that we are actually going to study further. That what is the protein that it is interacting to. It is indeed getting recruited to the SCV, but what is the D1 protein, because you have 40 odd protein there. So what is the protein that it is interacting with?
Thank you, ma'am. So I have one last question for you.
Yeah.
Rija, are there any other pathogens that evade the immune system just like Salmonella?
Are there any other pathogen that evade the immune system just like Salmonella? I will give you a dozen pathogen. The first pathogen that evade, of course, Salmonella evades the immune system, inhibits the antigen presentation, and that is the reason you do not have a protective immunity. You need this antibiotic intervention there. Then you have something known as, all of you know, Mycobacterium tuberculosis. Extremely, clever pathogen and strategic pathogen, I would say. For two reasons. First of all, extremely slow growing, very slow growing. And secondly, they also downregulate antigen presentation. And they are, in the sense they are very funny, because there is no difference in the interferon gamma level. If you see in the person who is being infected with MTB versus the one who is infected with any other, say pathogen XYZ. Now, the MTB infected individual, they have high level of interferon gamma, which shows that interferon gamma should have in fact cleared the pathogen now, but it does not do. Why? Because the interferon gamma receptor is downregulated in MTB infected individual. So though the body is making tons of interferon gamma, it is a futile exercise because there is no receptor to sense it. So indeed, this is one pathogen, one of the pathogen that now I am thinking, which is, acting, and it's a devastating role that it is taking, especially when you see it in the diabetic wound infection, is a combination of Pseudomonas aeruginosa, Staphylococcus aureus, and Acinetobacter baumannii. Now, Acinetobacter baumannii for a very long time was known to be simply lying around and doing nothing. But now we have figured out that how dangerous this is. And, when Akshay does his wound infection models, we exactly know that how this particular sneaky pathogen is sneaking in and causing a deep tissue invasion with the help of Pseudomonas and Staphylococcus aureus. So what you find in the deep tissue in the end is a Acinetobacter baumannii, causing a total tissue damage and the necrosis and unhealing, non-healing diabetic ulcers or the non-healing diabetic wound. So yes, there are, I will tell you that all the pathogens which are invasive, the example of a strict non-invasive pathogen is cholera, Vibrio cholerae, which does not have the ability to do anything other than releasing its toxin. And that is the reason once with diarrhea, when diarrhea sets in, the Vibrio also is wiped off from the system. Other than that, almost every pathogen that I deal with or I have come across have crossed this boundary of being extracellular, and they have gained that invasive property by utilizing many of these subversion strategies. And you do require really strict intervention strategies to get rid of them. Body by itself cannot evoke, in spite of having such a wonderful immune system, cannot evoke the response to get rid of the bacteria. Yes. How does the host immune system recognize and fight the vacuole inhabiting the Salmonella bacteria? Yes, trying to understand Tejaswini, but this entire Salmonella which is living in the vacuole, the host system, and it is a stealthy, so the host system do not recognize much in a much better way. But at the same time, the host system is also, not going to keep quiet. So few bacteria, initial bacteria, when it gets in, 5 to 6% of the bacteria, it indeed does cause a problem. It does evoke the response, but then the response is immediately shut off by the growing Salmonella numbers. And then by inhibiting antigen presentation, by downregulating certain genes of the immune pathway, it totally kind of shuts off the system. And they do not recognize. So it's a very stealthy approach. It's a very stealthy approach. That is why systemic infection caused by typhi and paratyphi, which is specific to human, must be, kind of avoided at all cost. It is no longer a simple typhoid fever, which was 100 years old. It is now gained a potency to travel across or to disseminate across the barrier, across the intestinal barrier. They become extraintestinal, and that is the worry. Right from endocarditis to meningitis to osteoarthritis, and crossing the blood-brain barrier. So it is no longer a very simple disease at all.
Thank you, ma'am.
Yeah.
Thank you, ma'am.
Thank you.
Unfortunately, we have a time constraint today and we cannot take up any more questions. However, we may convey these questions to our respected speaker if she permits. I now request Dr. Anija Philip to propose the vote of our respected speaker has put up her email ID in the chat box. And if anyone has any questions, you may direct those at the email ID that she has so kindly put up for us. Thank you.
That was indeed great question answer session, Dipshika. I'm sure the students have several more questions. We have a lot of them in the chat boxes and in our WhatsApp groups. So we'll be mailing them to you so that they are answered later on. It was
Sure.
indeed a very great session. So
Sure.
I
Yeah.
Yeah. So I request Dr. Anija Philip to propose the vote of thanks. I think we are not able to hear Anija. On behalf of Hislop School of Biotechnology and on behalf of the Department of Chemistry, I would like to take this opportunity of proposing this vote of thanks, dear Dipshika. Yeah, it was indeed a very great session. And on behalf of Hislop College, Hislop Education Society, and the entire organizing committee, I would like to extend a heartfelt thank you. And we are grateful to you for having taken out time and addressed the questions and also told the students about the exams. And as I have been telling my students always, that Dipshika madam is one person who would always invite you people. If you have a spark, she would invite you definitely to ISC for carrying out PhD and for internships. So thank you once again.
Thank you. Thank you all. Thanks a lot, Sunita. It was indeed a very, very interesting question answer session. And I would like to really thank you and everybody from Hislop College for giving me this opportunity. It was indeed my pleasure. And I indeed look forward for many more interactions. Thank you.
Yeah, sure, sure.
It's a request to all the participants to fill and submit the feedback form via the link in their WhatsApp group. The certificate of participation is linked to feedback form and would be provided to you in a few days. Thank you all for attending the webinar, and goodbye. Thank you.
I would now request the participants to leave the meet. Thank you all for joining us. Good day. With the festival season ahead, a very happy Diwali to all of you. Happy and a